to NWCVT 2020-201 organized by Dr. V. Sunita, Professor of Zoology, and also Dr. Dakshayani, Assistant Professor, Genetics and Genomics, and our Honorable Vice Chancellor, Professor Surya Kalavati Garu, respected Registrar, Dr. Professor Vijayaraghu Prasad Garu, and PRO Venkat Subbareddy Garu, and my other technical colleagues who are here. This is indeed one of the need of the day for the country and also for the globe. We know very well there were lots of pandemics were happened in the history of the mankind. So we battled them and we came back to the normalcy after losing few thousands to few lakhs of people. The COVID-19 recent past is making such a havoc in the entire globe. However, if you see this spread of the COVID-19 is more vulnerable to all the countries only due to the human migration and also the more advancement of the scientific and medical and other facilities. Though the medical facility was so advanced, but the death rates were unable to control due to the gravity of the COVID-19 pandemic. And in India, in particular, if you see in India, the problem is we are having the NMAS. And though our governments are doing extremely well, but it was not up to the level because of the NMAS and also the uneducatedness and also poverty is predominating than the health. So this is the really wonderful occasion. Our colleagues has taken the COVID-19 vaccination therapeutics, and also the two eminent resource persons, Professor M. Hema Garu, Professor and Head Department of Virology, Sri Venkateswara University, Tirupati, and Dr. Pratap Garu is Assistant Professor in RIMS. So they are where? RIMS Kadapa. So these two are going to emulate the things to be done after the post-COVID activity. So hopefully, we know now India is already vaccinated nearing to 3 crore people. So still we are being a 130. This number is so low. So still we are contemplating, we are feeling, we are predicting that the third wave may come. So we have to be more careful today, only as said by the government and also the standard operating procedures of the central and state government. Only three things must be always we should remember cautious. One is the cleaning of the hand with soap or sanitizer, and another one is the keeping the mask and social distancing of the 2 meters. Whenever your turn comes, you have to be vaccinated. Vaccines are good. There is no way that one is superior, one is inferior. So please, I am very much thankful to the organizers and also the administrators giving me the welcome address. Thank you, one and all. Thank you very much. Now, I request introductory remarks by our registrar, Professor D. Vijayaraghu Prasad Garu.
Warm good morning to all of you, Vice Chancellor Madam, our beloved Principal, and foremost, and all the participants. First, I would like to congratulate these two young scientists here from two different departments coming together, putting hand on hand, and bringing up a wonderful topic for today's discussion. And also, it is worth to note that they have brought two diversified people also, one from the basic research, Professor Hema Garu, and also another person, clinician, on board for today's webinar, which is a very fascinating thing. Interestingly, the topic is twofold. The webinar topic is therapeutics versus vaccines, if you say that way. It's a very interesting way to look at it, because there are a lot of, as we speak also, you would have realized, and there are a number of drugs or vaccines, they are hitting the market, bypassing number of regulatory, steps that need to be taken, which are very essential. But such is the psyche of the different people across the nations and worldwide. It's so fearsome that a lot of regulatory controls have been bypassed and they are hitting the market. So a lot of drugs have not been really tested. However, if you look at the vaccines that today that are available across the world, not just in India, which are nothing but Covishield, BioNTech, Pfizer, Moderna, and Sputnik, whatever, they are all subunit vaccines which are targeting this spike protein, right? Whether that is presented through a vector or it is through mRNA form. They are all aim to produce a protein which is ultimately going to produce antibodies and perhaps also proper T cell responses, which are essential for a good, proper immune response. While if you look at this Covaxin, which is only made in India, it's also a different type of vaccine, but it is a complete virus which is inactivated and it is administered. So a lot of people feel that it is one of the best way because you have a lot of epitopes being subjected for getting a better protection. However, I think that today's speakers will enlighten us on more about these two things. While coming to the therapeutics, it's most debatable and widely criticized because they are all repurposed drugs. Repurposed means they are not actually meant for COVID protection. They are actually used for different clinical aspects while they are repurposed for COVID clinical purpose. Here, if you look at these drugs like doxycycline, ivermectin, or corticosteroids, doxycycline, if you look, it is not antiviral, it is just antibiotic, means antibacterial. It is widely used for a number of skin therapies in clinical stages and also urinary tract infections, scabies, and other places. While, very interestingly, if you look more carefully, it also is a matrix metalloprotease inhibitor, which is MMP2 inhibitor, because the lung architecture is very delicate. It's by collagen and elastin. These two bind all these L1 cells, lung L1, L2 cells, which imparts this lot of elastic property. Because this doxycycline is, as I said, these are all repurposed drugs, where this doxycycline can inhibit this protease activity, which degrades the lung architecture, is being protected by doxycycline. But it's no antiviral activity, but it protects to some extent. So therefore, a lot of research, ivermectin, if you could, it is an anti-helminthic, nothing to do with any antiviral activity. Helminth means, you know, these pinworms and other things. So corticosteroids, global suppression of immune responses. So people are using at will and, you know, it's like free fire. Therefore, a lot of research and enlightenment is required as to how to move forward and use these things in a very more judicious way and bring some sense at the end of the day. I do hope this one-day webinar will enlighten us, all of us, on these aspects. I thank the organizers for giving me this opportunity to speak over here. Thank you very much.
Second class present. Second class.
Thank you, Dr. Professor Prasad, for giving the introductory remarks. You have thrown light on the medication versus its effects for the human organs. And really, you have enlightened the theme of the today's national webinar. And now it is time for us to listen our Honorable Vice Chancellor, Professor M. Surikalavati Garu. Actually, I have to say only few words about her. She is a very good human being and very, very, very kind-hearted person and always cheerful. Now you can see in the
cheerful people don't get affected.
And also, as my registrar pointed out, cheerful people may not be affected by the COVID-19. So, Madam, now I request you to please deliver the presidential address, Madam. Thank you.
Thank you.
Please, Madam.
Thank you. So, good morning, respected Registrar of the University, Vijay Raghav Prasad, the Principal, Krishna Reddy Sir, Sunita and Dakshaini, our PRO Subbareddy Garu, and participants and today's speakers, Professor Hema Garu and Pratap Garu. Good morning, participants. Good morning once again. So first I want to congratulate Dakshaini and Sunita for organizing from two different departments. They are organizing this national webinar on COVID-19 therapeutics and vaccine. This is a very relevant nowadays because already second variant is coming, and that's why it is very, very important. So this coronavirus disease, so that is COVID-19 is an infectious disease caused by newly discovered coronavirus. Most of the people infected with the COVID-19 virus will experience mild, moderate respiratory illness and recovery without requiring special treatment. So scientists around the world has emphasized that vaccines along with the therapeutic support are the only effective tools to contain and the ongoing COVID-19 pandemic, severe acute respiratory syndrome, coronavirus 2, that is SARS-CoV-2 is the coronavirus 2 is the causal agent of COVID-19. Scientists have developed or are in the process of developing different types of vaccines which differ greatly in terms of their basic design. Since the outlook of the novel coronavirus disease, COVID-19 caused by the SARS coronavirus 2, this disease has spread rapidly around the globe. And considering the potential threat of pandemic, scientists and physicians have been racing to understand this new virus and the pathopsychology of the disease to uncover possible treatment and regimen and discover effective therapeutics agents and vaccines. So this webinar is based on the awareness of COVID-19 therapeutic and vaccines. So you may get benefit from these things for dear participants. So thank you for giving me this opportunity.
Thank you, madam, for enlightening the theme of the today's national webinar. We are very fortunate to listen the importance of the two themes, vaccination and therapeutics for the COVID-19. So now I call upon Dr. L. Dakshaini, Assistant Professor, Department of Genomics, to introduce one of the invited speakers, Professor Hema Garu, Department of Virology, SV University, Tirupati. ఇంటూ పెట్టండి మా ఇక్కడ అదే రాసింది పెట్టండి చెప్పండి అబౌట్ ది సెమినార్ చెప్పండి ఓకే ఓకే అమ్మా సో సారీ దేర్ ఇస్ అ స్మాల్ చేంజ్ ఐఎమ్ సారీ ఐ కుడెంట్ రీడ్ ప్రాపర్లీ సో నౌ ఐ కాల్ అపాన్ డాక్టర్ దాక్షాయిని గారు టు ఇంట్రడ్యూస్ ది webinar.
Thank you, sir.
Please.
Thank
Very good morning and welcome to today's national webinar on COVID-19 therapeutics and vaccines. The world has been in varying states of lockdown since March 2020, causing profound economic and social consequences. In addition, despite implementation of masks and other safety protocols, a number of lives have been lost to COVID. COVID quickly and unexpectedly spread nationally and internationally, with millions worldwide already exposed to the virus. Some estimates indicate that between 1 to 20% of the population have been exposed to COVID-19. However, these estimates are questionable as many patients are asymptomatic and go untested. Unfortunately, immune response to a pathogen is often heterogeneous and varies between individuals based on age, environment, and underlying health conditions. Preventive vaccination is the safest and most cost-effective way to prevent COVID-19 illness and death, and the best option to combat anticipated future variants. A similar global strategy is warranted to prevent or minimize COVID-19 and future SARS coronavirus 2 variant prevalence. COVID-19 is an enormous scientific, clinical, and social challenge that innovative vaccine formulations play critical roles to overcome disease. Scientists around the world have emphasized the vaccines along with therapeutic support are all only effective tools to control the ongoing COVID-19 pandemic. Today's webinar is right in time to discuss about various strains and available treatments. This webinar is aimed at providing an overview of the efforts, available treatments, and effective vaccines for this novel coronavirus. It has gripped the world in terms of economy, human health, and life. And today we have two speakers. We have very few virologists in our state, and we are lucky to have the Professor Hema is one among them. She will speak on coronavirus, and Dr. Pratap, Assistant Professor, RIMS Medical College, will speak on the management of COVID-19.
అయిపోయిందమ్మా గుడ్ మళ్ళీ మీరే కదా వినపడుతుంది సో నౌ ఐ రిక్వెస్ట్ అగైన్ డాక్టర్ దాక్షాయని గారు టు ఇంట్రడ్యూస్ ది ఇన్వైటెడ్ స్పీకర్ ప్రొఫెసర్ హేమ డిపార్ట్మెంట్ ఆఫ్ వైరాలజీ.
Very good morning again. I am very much pleased to introduce our invited speaker, Professor Hema from Department of Virology, Sri Venkateswara University, Tirupati. Ma'am completed her MSc in 1992 and PhD in the year 1998 from the Department of Virology, SV University, Tirupati. Ma'am worked as CSR RA from 2000 to 2002 in Virology Department, Sri Venkateswara University. Ma'am did her postdoc from 2002 to 2004 in the Department of Biophysics and Biochemistry, Texas A&M University, College Station, Texas, USA. Ma'am worked in Indian Immunological Limited in the R&D from 2004 to 2006. Then ma'am joined as an assistant professor in the Department of Biotechnology, Sri Padmavati Mahila University, Tirupati in the year 2006. In 2009, ma'am received DBT Overseas Associate in the Department of Cellular and Molecular Biochemistry, Indiana University, USA. Then ma'am joined as associate professor in the Department of Virology, SVU, Tirupati. And in 2015, madam joined in the Department of Virology as associate professor and promoted to professor in the year 2015. Ma'am has 5 patents as first inventor, India, USA, Australia, and Europe. She has published 55 publications in reputed national and international journals with high impact factor. She received 6 major research projects from various funding agencies, UGC, CSR, and DBT. She received United States fellowship, that is USIEF, to conduct the virus-based nanotechnology research at Case Research University, Ohio, and USA. Ma'am received European Anonymous Mendes Brave Visiting Professor Fellowship to conduct research on the RNA-based resistant at Agricultural University of Athens in 2017. Ma'am is the fellow of Andhra Pradesh Academy of Sciences, Amravati, India. And I am heartily welcome Professor Hema, and I am sure that your presence make this webinar a very successful one, and we shall be enriched by sharing your ideas and experiences. And I request Professor Hema to speak on this occasion.
On the mute bit.
Thank you, Drakshayani. I express my heartfelt thank you to Professor V.C. Madam Garu, Professor Vijayaraghav Prasad Garu, Registrar of the Yogi Vemana University, Principal Garu, and the organizers. I'm really thankful for this wonderful opportunity, and which is very apt for the situation. Am I audible?
Loud and clear.
Are you able to see the screen, Drakshayani?
Yes, we can see the screen.
Okay. As the organizers and Vijayaraghav Prasad Saru said rightly, that there are a lot of different informations that are available, and everywhere we are surrounded by so much of different ideologies and variety of information that is available with respect to the media and the newspapers, and then everywhere in the scientific journals, tons and tons of information is available, and we really don't know which one is authentic, and there is no time for anybody to look at its authenticity, all that is going on around us. But still, I tried my best to get the scientific information from the journals, very authentic journals, and I'm trying to present it. So as I know that the next speaker, Dr. Pratap Garu, is going to take care of the management of the disease. So I thought that, as you know, that for control of any virus, you need to have the information about the biology of the virus. So unless otherwise we know how it is going to infect the cells, I mean, how it is going to enter into the body, what is that it is doing inside the body, we will not be able to get to the control measures. So it is very, very important to know about the virus in detail. So that is still we are lacking with so much of information, but whatever information it is available, I thought that I'll just go overview about it. Okay. So as organizers said, I'm from Venkateshwara University, a NAC A+ grade university. And then I'm going to give an outline of the things here in this particular presentation, where I'm going to cover the origin of the pandemic, and then structure and pathogenesis of SARS coronavirus 2, and then I'm going to cover a bit of diagnosis, and then transmission and prevention. And basically, I'm going to cover a lot of information with respect to the vaccines and therapeutic interventions. And I'll be happy to answer all the questions from the participant, and that will be much more important than the original session, actually. So just to have a glimpse of an idea about the virus structure to the first year students, if at all, if they are there, anybody, as you know that viruses are simple entities. We cannot call them as organisms because of the reason that they don't have any kind of organelles that you see with respect to other pathogens. It is very simple. Generally, it will have an inner core where it will be having its genetic material. Either it can be RNA or DNA, but you will never have both. And then it will have an outer capsid, which is usually made up of capsid protein, or you can have some more structural proteins. And there are some viruses which are complicated, which will have envelope also. It can be made up of either carbohydrate, protein, or lipid, or mix of all those entities. So I've given some examples also here. So just to give you one more idea that, so the viruses are able to enter into the cells through receptors, cellular receptors. And once they go inside the cell, they try to make many, many more copies of them, and then produce tons of virus particles, and then they get released from the cell, and then they go infect the other cells, adjacent cells. So while doing that process, actually, so what they do is that they try to hijack the cell machinery, because as I said just now that viruses doesn't have any kind of cellular machinery, so they need to take help of the cellular machinery, and the energies, and then DNTPs, and all kinds of different aspects of ribosomes especially, and then all kinds of different cellular machinery will be taken care and hijacked by the virus, and it will try to make sure that it will use for its replication and make many, many, many copies of it. So what we understood just now is that viruses are able to enter into the cells on their own, and they could able to multiply, and by hijacking the host cell machinery, and they will try to take all the cellular machinery for their replication, and then they can make more copies of it to infect the adjacent cells. So we are trying to go through this word pandemic. Everybody is calling this situation as pandemic situation. So what is pandemic? Pandemic is nothing but the worldwide spread of the new disease. So if at all, if they spread a new disease is spreading all through the world, so then that situation is called as pandemic. And if it is affecting significant proportion of the population, then again it considers under the pandemic. So it is not, as Principal Sir said, we are not new to the pandemics. We have gone through many pandemics, both big and small pandemics, all through this eras, if you look at it. So I've given a glimpse of it, and we have gone through a death toll with respect to several numbers also. I'm not going through the details of each and every one, but by looking at the picture, you can understand that there are small and big pandemics we have gone through, and we have come across all those problems also within no time. So we just have to have that hope that we will be okay with it. So when you look at the SARS coronavirus, so the classification, if you want to look at it, so it belongs to order Nidovirales, and there is a family which is called as Coronaviridae, and under the Coronaviridae you have two subfamilies. So the coronavirus belongs to the Orthocoronavirinae subfamily. So under the Orthocoronavirinae you have four genera. So under the four genera, so SARS coronavirus belongs to the beta coronavirus. So both alpha coronaviruses and beta coronaviruses are zoonotic diseases. They cross through some of the animals, and from the animals they can come to the human beings. So that is why I have highlighted these two things. So these are very important, especially beta coronaviruses. We have significant number there are almost 7 viruses, alpha and beta coronaviruses, which can infect the human beings, and that's where we get into this whole problem right now. So we have a list of alpha and beta coronaviruses, and among all these 7, the 3 important coronaviruses, especially the MERS coronavirus and SARS coronavirus, or coronavirus 1 and SARS coronavirus 2, all 3 of them are beta coronaviruses, and they're almost having more significant homology. So that is why we have to look at these 3 things a little more details. So when we look at the history, the SARS coronavirus 1 or SARS coronavirus, it has come in 2002 and 3, and it is also coming from China only, and it has come from bats to civet cats and then to the humans, and so the death rate is 9.6, which is high, and 8,000 total cases and 774 deaths. So comparatively, we think that it is less number, but death percentage is very high. Then we come across, after 10 years of SARS coronavirus 1, we got into MERS coronavirus, so which is in 2012, so which has been originated from Saudi Arabia, and then again the death rate is much more high, 34.4%. So though we have 2 situations in before, but we could able to come across these situations, and we were able to curtail these 2 diseases because of the reason that when compared to the present situation, why we don't have so much of problem with respect to these 2 viruses, because of the reason that so they could not spread to the level that the present virus is spreading. So they don't have, so though you have SARS virus 9.6% of death rate, but it is not spreading as it is spreading as the present virus. So that is why we could able to control it, and the efficiency of binding of this virus to the human cells it is not so strong as the present virus. So that is why we could able to control by having different control measures, but we are not able to do that with respect to the present virus. So the history of the present virus, I'll try to deal with it a little later. So I just want to tell you that, so these viruses, human coronaviruses especially, they are going through some of the avian hosts as well as mammalian hosts like snakes, bats, tigers, civet cats, and mice, dogs, and pangolins. So that is why it is a zoonotic disease, and it is much more important when it comes to zoonosis. So when you look at the origin, you all have already so much of information. I just got some dates from the literature. So the first case has been identified in November 2019, and then fever case especially, it has been recorded in hospital in December 2019, and 31st December, China has reported to WHO saying that there is a problem in their country. And then, so in January 2020, so the man who has come from Wuhan state, so he was identified with disease in US. And then Chinese government also has declared that they have spread up the disease. Human to human transmission has been confirmed both in China as well as in US. So then, so the coronavirus has been announced as a pandemic by WHO in 11th March. So then US President declared a national emergency on 13th March, and India has gone to the lockdown from 24th March onwards. So after that, we are going through this situation with continuous lockdowns and then different other problems, actually. So as already organizers said, that we have a greatest global crisis that ever happened, and the coronavirus is increasing in alarming rates and threatening the whole world. So we are not still got rid of this problem. We are still in the problem. And then it has created an unprecedented demand for healthcare. So this is a major situation where we have a lot of demand for the healthcare, and especially healthcare system in some of the area, some of the country, it has completely collapsed, and it has given a lot of problem for the non-COVID patients. So, and it affected global economy all through the trade, travel, education, and there are many people affected by both physically as well as mentally. So this is something different situation what we have come across before when you really look at it. So there are numbers I've given as of today. I mean, yesterday I've taken these numbers from the WHO site. So the numbers are huge. And then the disease is, as I told you, that the COVID-19, the disease name is COVID-19, but it is caused by SARS coronavirus, severe acute respiratory syndrome, coronavirus 2. So that is what it has been named by the WHO. So when you look at the incidence of COVID-19, so we have gone through a severe, disease and a rise in the cases in 2020 from April to July and August. And then again, this year we have got a huge peak compared to the previous one. It is much more higher. And then so when you look at the statistics a little more, so this information has been taken from an authentic site, Our World in Data, which is allowing all of us to take the charts from there. So this is a cumulative confirmed COVID-19 cases. You can see the peak rise still, and it is not coming down. And then daily confirmed cases, slowly it is coming down, but still we have a huge peak when compared to the previous year. So when you take the cumulative confirmed deaths with respect to COVID-19, I've taken the other countries also here, and cumulative confirmed deaths this second wave we have high numbers, and then the newly confirmed deaths, so we are okay, we are still getting down, and that is a good thing actually. So confirmed cases, again, if you look at the world map altogether, so the dark color indicates the severity of the problem, and then we are also getting into darker region, which is not that great actually. So the global confirmed cases and the vaccination status also I've given with respect to the numbers for yesterday date. And in India, we have got 1.2% deaths, so which is okay, which is not as severe as US and other European countries, if you think about it. But with respect to vaccination, we are still 37 crores, and that is not enough. So we, our population is almost 140 crores, and we have to go a long way. with it. So now I'm going to get into the real picture of virus structure and many other details about the virus. So we have looked at the problem, and we know that it is a problem if the cases are increasing and it is going down and getting up again and again. So to control this situation, we need to have complete information about the biology of the virus. So as you see the virus structure altogether, so it looks like a ball here, and because of the spike proteins which are present on top of it, it looks like a crown. So when you see the cross section, it is looking like a crown altogether. So corona means crown. So Corona Viridae means, so that is how it has got the name as such. So when you look at the structure, as I told you that every virus will have a structure, a capsid structure outside. So this particular virus has got 4 structural proteins. So the spikes are present on top of the transmembrane envelope, and then those are spike proteins, and then it has got a nucleocapsid protein which will be embedded into the nucleic acid, and then it has got a membrane protein which is present on the envelope, and then it has got envelope proteins between the membrane protein and the spike protein. So it has got a single-stranded unsegmented positive sense RNA as a genome. So that is anyway it is present, and which is very, very, very long. So when compared to other viruses, this particular virus is very peculiar because of the reason that it has got a long RNA genome. So one has to remember this because it is going to have a lot of impact in the control and prevention. So as I told you that it is enveloped, non-segmented, positives and single-stranded RNA is the genome, and approximately 26 to 32 kilodaltons depending on the type of coronavirus it is. And the 4 structural proteins are given here, and which are having different functions with respect to it. And this is the original electron microscopic picture. The viruses has been colored with blue. How it is going through the lung cells, it has been given here, the original picture actually. So approximately 70 to 80 nanometer size, and then the helical nucleocapsid, which is inside, you have the nucleocapsid proteins altogether, and the S protein is divided into 2 domains, S1 and S2. S1 head is the one which is having the receptor binding domain, and S2 is the anchor onto the lipid envelope so that it will be able to help for the fusion of the virus into the cell. Okay, so then we need to get into the transmission. As you know that the virus is basically aerosol, so it is entering into the body through the nose and mouth mostly, and then directly it is aerosol, because the aerosols, the droplets which are containing in the aerosol will contain the virus, and then when people are inhaling the aerosols, they get this problem inside, and then it is present very active in the surfaces also. That is how it is indirectly helping and getting into the body. And then contacting subjects also is a problem again, doors, elevators, and all the other objects. And if at all, if you touch the eye, nose, mouth, so this kind of different issues with respect to the transmission is going to help them a lot for the spread of this disease. So primary route is generally airborne. So because the way virus can survive for a long time, and then secondary route is contaminated objects. So different objects also have given how long the virus has been active on different things. So when you look at the genome organization of the virus, so then you have, as I told you, that it has got a long genome. So I'm not going to go into the details of genes and the thing, but I just want to tell you that the genome is well organized with the different ORFs, and there are almost 16 non-structural proteins are present and 4 structural proteins are present. Just now I said the spike protein, envelope protein, and membrane protein, and nucleocapsid protein. So it is well organized, and it has got all the inputs that are required for its replication. Whatever it is required from the host cell, it will anyways take it. And for that also, the viruses are coding for genes which can hijack the cell machinery. So that part is also available here. See, for example, I can tell you one thing here, one interesting thing actually. So this is an RNA virus, and it is coding for endoribonuclease. So endoribonuclease is a RNase. So it can chew the RNA. So you must be wondering that, see, when it is an RNA virus, when its genetic material is RNA, why the hell it is having the RNAs? Because if it is having RNAs, then it can cleave its own RNA, right? But it is not doing so because it is going to modify its RNA such that this RNAs will not work. But then why it is having RNAs? Because it wants to chew the host RNAs and take those precursors, ribonucleotides, and then use it for its replication. So such a smart virus it is. Such a intriguing virus it is that it has got all kinds of important genes which are coding for different important functions, and so very powerful actually with respect to it. So when you look at the comparison between the three important SARS viruses, MERS virus and coronavirus 1 and 3, so it is looks like similar, but there is some differences with respect to the genome organization. So when you look at the SARS coronavirus 1 and 2, you will see a lot of similarities, but MERS coronavirus is little different. So between 1 and 2, you have almost 78% similarity, but MERS and the SARS coronavirus 2, it is having only 50 to 60% similarity with respect to the genome sequence. Because why we need to know about all this information, because as we are undergoing a situation where we don't have time to look for new vaccines and new therapeutics, we are using the repurposed vaccines and therapeutics which are working for which people have worked for MERS and SARS. We are trying to use them only for the same strategies only we are using for now control of SARS coronavirus 2. So we need to know what is the similarity between those viruses also, so that how best they work for this virus also, we will get an idea. So then just to give you a glimpse of idea how the virus is entering into the cell, especially the different lung cells and then many other cells which are having the ACE2 receptor. So ACE2 receptor is angiotensin enzyme receptor, which is present in many of the cells, the epithelial cells of many of the vital organs actually. So when we will look at the detailed structure of the spike protein here, structural domains of the spike protein. They have different domains, but let us focus on the orbit. Exactly. Just a second, okay. So it has got a domain, receptor binding domain. And as I told you that S1 domain is the one which is having the receptor binding domain, which will be binding to the ACE2 receptor through the help of S2 domain. Okay, so S2 domain is the one which is helping for the fusion of the S1 domain to the ACE2 receptor. So that is what it has been explained here. So there is one protease, host protease, transmembrane protease, serine protease, which is very, very important, and which will try to cleave this S protein S2 domain, and which will try to help the S1 domain to bind to this ACE2 receptor. So this host protease is also important. So we need to understand this whole mechanism of entry. So once we understand this mechanism of entry, what are all the participants which are participating in the cell fusion, virus fusion into the cell, then we will be able to curtail this particular steps. We can stop these steps through identifying some drugs or therapeutics which can stop these things. So we need to understand that there is a protease which is helping, there is ACE2 receptor which is acting as a receptor for binding of this RBD domain of S1. So all this is important. So this is again from WHO site I have taken, where viruses are the yellow balls are viruses. How many viruses are infecting the lung cell, you can see in this picture. So then the other thing I want to highlight, bring it on to your attention, is that so ACE2 is present. So why the cells are having ACE2? So ACE2 is present for a particular function where it can balance the signal transduction pathway of this RAS system. Okay, so ACE2 is a key regulator of this RAS system. So I'm not going into the details of all this signal transduction because it is not the subject of today's seminar. So it is going to balance the RAS system and the signal transduction. So that means that if it is blocked by the SARS coronavirus, then there is a chance that the balance is disturbed. So generally it will try to take care of the anti-inflammation, vasodilation and antioxidant effects. So if ACE2 is normal without affected by the SARS coronavirus 2, then it is going to take care of many good things. So if it is blocked by the coronavirus, and if the coronavirus is trying to enter through it and it is going to block this side, then there is a problem that you can have vasoconstriction, and then you have aldosterone secretion, increased H2O absorption, and then endothelial dysfunction. So these are all problems so that are getting into an inflammatory state. So that is what it has been explained here. So there are two problems, like the virus itself is going to create some problems after entering into the system. But before entering, if it is blocking the ACE2, because ACE2 is important for many good things, if it is going to get disrupted, there are bad things that are going to happen, that may happen. So we need to understand that where are the organs we have the ACE2 expression. So the recent study is showing that there are several vital organs that you see in the heart, and then some of the liver cells, and some of the lung cells, and then kidney cells and the spleen cells. There are many different organs, especially GI system also is having the ACE2 expression, RNA expression, and in some of the cells they have seen the expression of ACE2 protein receptor also. So that indicates that the COVID-19 is able to enter through not only into the lung cells, but also into the GI tract, as well as all the other important vital organs, and it can create some kind of condition here. So now in the second wave, we realized that the people are suffering with heart failure, brain death, and then there are the blood clots are happening, and then people are undergoing the vascular inflammation, and then anyway, first wave we have come across this acute respiratory distress syndrome anyways, and respiratory failure. But in the second wave, we have come across all these things because the variant has got a much more stronger binding capacity to the ACE2 receptor, and that's where it is trying to enter into the lung cells and all other cells in much more efficiency, with much more high efficiency, and it is able to affect many other things also. So when you look at the multiplication of the virus, so in the lung cell, if you look at one cell, what exactly is happening is that the coronavirus is able to bind to the receptor, ACE2 receptor, and through the receptor mediated endocytosis, it is going to enter into the cell, and through the fusion, it is going, when it is entering, it will release its RNA. So RNA will undergo translation and transcription, and then you multiplication, and you will get all kinds of many copies of the RNA molecule, and they undergo again translation and produce all the proteins. And all the proteins, once they go into the Golgi complex, they get into assembly. And then once the virus is assembled, it will try to get its membrane from the cell membrane, and then it will come out of the cell through exocytosis. So this is what is happening. So once the virus is entering into the cell, it will take just in hours to days. It will be able to produce many copies of the viruses in single cell. Like that, if many cells are getting affected, you are going to get tons of viruses. So that is again from the WHO site. So how the viruses are, the yellow balls are viruses in the lung cell, the alveolar infection has been shown. So that means that this particular, so people are asking many times that aren't we getting cold and fever all the time? So why this virus is so different? Why it is so specific? Why we are having so much of problem? Because of the reason that it is 10 to 50 times virulent than the flu virus. So than the influenza virus. So that is how the whole problem has come. And it has been considered under the risk group 4 virus. So risk group 4 is very top infectious virus. And then there is a high community risk. And again, the problem is that we don't have a specific treatment regime for this particular virus. So we are trying to treat the patients. Doctors are treating the patients based on the comorbidities, based on the situation that they are facing at that particular time. They are trying to give the treatment, but there is no specific treatment regime, so that we can take it and then we can say that, okay, by giving this treatment, the patient will be okay. There is no such situation still. So that is why it is getting into more problem. So before getting into the cytokine storm and other things, I just want to give you a glimpse of general immune response, just for the students' sake. Please, you have to bear with me. All the other professors and lecturers, though you have some information about this, but for students, I just want to give this information that, so we are surrounded by so many microbes around us. So there are many, many things. So you see helminths here, you see viruses, you have bacteria, you have fungus. So a lot of things we are, we are always surrounded by many, many, many microorganisms. So what our body does, so whenever there is a microorganism attack, then we have a immune system inside our body, which will be having both cells of the immune system, organs of the immune system, where they will have specific mechanisms to take care of all the pathogenic organisms which are going to enter into the body. So when you look at the whole immunity, you have two types of immunity. One is innate immunity, which is inherent, which is less specific immunity. Other one is adaptive immunity, which is a slow response, but very, very specific, very powerful immunity. So we understand that in the innate immunity, we have different types of cells which will take care of it. The pathogens when it is entering into the body as a first line of defense. But in the adaptive immunity, once it is entering into the body, after breaching the innate immunity, it will be able to enter, because pathogens are very smart. They know how to break this innate immunity. They know how to go inside the body. So once they are inside the body, then adaptive immune cells will come into the picture. They fight against the pathogen. They try to clear the pathogen in a way, in a different mechanisms altogether. But we need to understand that these two things are coordinated way they will work. So these innate immune cells will help the adaptive immune cells. These innate immune cells will help the adaptive immune cells in such a way that they'll be able to get activated and differentiated into effector cells. So one has to remember this aspect. And then when you look at it, as just now I said that, so for the activation of adaptive immune cells, the innate immune cells has to send their effector molecules like cytokines, which will be helping for the activity of these adaptive immune cells. So that means that the macrophages and dendritic cells and other cells which will be producing the cytokines, which will be helping for the B cell and T cell activation. So,
Hema, sorry for the interruption. Presentation is not seen here, visible.
Somebody has switched on their webcam, and that is why I lost it, I guess. Anyway, I'll just share it once again. I'm sorry about that, but I realized that somebody is switching on their webcam. Okay.
Yeah, we can say. Thanks.
Okay. So the point is that I just want to highlight here that the two innate immunity and adaptive immunity, the two important immune mechanisms are working together, complementing each other, the different aspects of their functions, and then they'll be able to help each other in such a way that they'll be able to get the activation and they'll get into the mechanism of action. So, but so there is one more side of the immune mechanism where, if at all, if it is overreacting, if at all, if it is reacting unnecessarily, when there is a pathogen, if innate immune system is working and adaptive immune system is working, that is fine. We will get rid of the pathogen and we will get the protection. But if it is unnecessarily, if it is working and if it is getting activated, then there is a problem. You have autoantibodies, you have more cytokines, which will be dangerous, because these cytokines, once they release, they don't be silent. They go and try to create a problem onto the major organs. They try to give the inflammation onto the major layers of the organs, and that inflammation can lead to the damage of those organs. And so that is why the dysfunction of immune system is again problem. So that means that it has to work in an optimum way. If at all, if it is going to go into a different level of action, if it is too much activated also is a problem. If it is less activated also is a problem. So why it is going into the hyperactive stage, then one has to realize that the virus infection, the cancerous growth. So these are all the conditions. Once the virus is under stress by virus infection or by some changes in the cell structure, cell physiology, then there is a chance that they go into crazy, and then they go into the receptor mediated interactions, and then they activate the immune cells so much, and they get release of so much of cytokines, which is going to be a problem. So that is what is happening with respect to the coronaviruses also. So what exactly is happening? So it is trying to activate the TLR pathway, and then the transcription activators are binding onto the DNA, and they produce more and more of cytokines. So cytokines, especially interleukins and interferons, and then the TNF. So these are all the cytokines which are protective at the optimum levels. But if they are producing too much, then they go and bind to the other cells, the innate immune cells, and they activate more of these cells, and again so much of interferons and other inflammatory cytokines will be produced. So these cytokines in an unregulated amounts, if they have, they go and bind to all the other vital organs, and they damage all these different organs. So how they do it, we will see it. So these are all the different pathways which are showing that how the cytokines are produced through the coronavirus infection. So if there is a list of cytokines which are produced by the coronavirus infection, which can be produced. It is not all the time case, okay? It depends on the patient status. So cytokine storm will be a case in a particular situation. So where this is going to be a problem. So how it is going to take care of it, you just see it. So the cytokines which are released, what they do is that they activate all these different molecules, and those molecules will try to damage the tissues, and then it can lead to the multi-organ dysfunction syndrome, and it can lead to the deaths. So what are all the organs that can be damaged? You see it. So it can be lung, GI tract, and brain, CVS, central, then see liver, kidney, microcirculation in the blood circulation also, and in the eye also. So there is a different things that are happening with respect to the immune dysfunction because of the coronavirus infection. That is how the people are getting into the problem, and they are dying. Okay, so if you look at the cytokine storm and multi-organ damage, exactly is what is happening with respect to each organ. If you see it, so these are all the different parameters which are going down and up, and then that is how the multiple vital organs are going to get problem, and then people are suffering with all these diseases, and then it will lead to a problem. So where the acute respiratory distress syndrome, heart failures, renal failures, kidney failure, liver damage, and then multi-organ failure is also a problem, and people are dying with different problems with respect to the cytokine storm. So that is something has to be noted down, and there are different markers altogether where you can check all these different effects of SARS coronavirus, and oxygen levels will be decreasing because of this alveolar infection, and then blood clots. So you can look at the D dimer and the C-reactive protein also, you can look at it. So all these things. So through immunological effects, you are having all these problems, and non-immunological effects also you have problems. So especially if the brain is getting affected, then neurological complications also people are trying to get a clear picture on it recently in the recent publications. So when you look at the total COVID-19 cases, you will be able to divide them into 4 groups actually. Generally, it will be mild illness. So many of the patients are almost 70 to 80 of the percent of the people, they will go through only absence of symptoms or very light symptoms. So in some cases, you will have moderate illness. So we will have lower respiratory disease and throat pain, and you will have most of the times their oxygen levels are okay. Severe levels, you will see respiratory problems, and then you have less oxygen, less than 94% actually. And in the case of critical illness, it will be generally multi-organ failure and septic shock, and then respiratory failure will be a problem. So that is how you can group the COVID-19 cases. So here also, if you see it in the pathogenesis, if you look at it, so pathogenesis is nothing but once the virus is entering into the system in the body, so what are all the stages that it goes through? So in the phase 1, the infection of epithelial cells can take place, and then in the phase 2, the acute inflammation will be a problem because of the immune cell too much hyperactivation. So pro-inflammatory cytokines will try to create the inflammation on the vital organs. So because of the cytokine storm that you see, and in the severe cases and resolution, so there is a acute respiratory distress syndrome, shock, and all the multi-organ failure can take place. So generally, as I told you just now, the 4 groups of cases. So incubation period will be 5 to 6 days, but it can range to 1 to 14 days. This is again a problem. See, we need to look at the COVID-19 in different perspectives actually. We cannot just say that this is it, because we are still digging into so much of information. We are getting tons and tons of information every day from the scientific, authentic journals, and the people who are working at the global level. People are working day and night to get more and more details about this virus. So we need to go through, we need to have an idea about all these things time to time. So initially it was thinking, people are thinking that it is just 5 to 6 days, but it is now ranging 1 to 14 days. Even the virus is also able to multiply for all these times, and symptoms also able to show all these 14 days. And most common symptoms are fever, dry cough, and fatigueness. But less commonly you will see the headache and hemoptysis and diarrhea, and disease progression can be leading to the pneumonia and acute respiratory distress syndrome, and sometimes heart failure, and then leading now the black fungus and other diseases are getting into picture, where secondary infections, because people are getting steroids to lower the immune function, which is hyperactive. So because of that, the normal pathogens which will not cause the disease, but they are getting into hyper, and then they are creating problems. And that is what it is now with respect to the black fungus and other yeast infections. So then, who gets severe disease? So if you look at the risk factors, so age, sex, obesity, comorbidities like BP, diabetes, and malignancy can be a problem. And genetics, viral factors, and immune factors all can be risk factors. And people who are in high age can be of high risk. Generally, that is what it is. Health condition also, who are having complications will be of high risk. Sex, somehow females are less susceptible to the disease than the male. It could be because of the genetic factor or viral factor. We really don't know at this point. So if at all, if you look at the symptoms and spectrum of symptoms, so most common symptom is fever, and then the percentage I've given, the atypical symptoms and typical symptoms. So that is how, again, taken from an authentic source here. So then spectrum of severity. So as I told you already that there could be shortness of breath and ARDS. And the clinical symptoms and signs also, you will have the D-dimer levels can be increased, and C-reactive protein can be increased. So these are all the markers that you can use for testing also. That is why we need to have this information. And you have the early infection, pulmonary phase, and hyperinflammation phase, where you get into a dangerous situation. So that is what it has been explained here again. And then when to seek care, when you need to be very careful with respect to the symptoms, and whenever you get into a problem of severity, then you need to go for health care, and then you need to call your primary doctor, self-isolate. And if at all, if you're suspected by exposure, you need to go for self-isolation, and it can affect anyone and everyone. But elderly are more susceptible, and more men are susceptible, and high risk, or all these risk factors I've given already. Okay, so the variants, if at all, if you want to look at the variants, so now people are talking about so much about variants, telling that Delta variant and some other variant, UK variant, all that. So I'll try to give you an idea what is exactly a variant actually here. So we understood that already the coronavirus genome organization is very complicated, and it has got a long genome, and compared to all the other virus diseases, is very, very long genome it has got. So if you look at the genome and especially the spike protein, if you look at the structure, so you look at the different variants, the spike protein structure, where the recognition motif is getting changed. So in the multiplication time, RNA-dependent RNA polymerase able to enter errors, and it will be able to tend to add the wrong nucleotides. So that's how you get the mutation. So whenever there is a mutation in the sequence, so there is a change that the structure of the spike protein may change a little bit. So then you must be asking a question, then what will happen if at all if a structural change takes place in the spike protein? So the change can lead to a problem that the virus may become more efficient in transmission. Either way it can happen. It can become less efficient or more efficient. But viruses also like us only. So they try to always struggle for their existence. They try to change themselves so that they'll be able to infect human beings more effectively. They try to evade and they try to make sure that they exist. They try to live all the time inside the cells. So they will live only once they are inside the cells. So they will make sure that they will do that more efficiently by changing themselves. So that is what is happening here in the spike protein. If at all if it is changed, then the efficiency of the binding of spike protein to the ACE2 receptor may change, and it can lead to a highest transmission. So that is one problem. The other problem is that if at all if the genome is changing, there is a possibility that it may multiply even more efficiently and produce more and more copies of the infection. It can become more virulent. And that is also a problem. More transmission, more multiplication, everything more is a problem here. That is how we are getting into the situations, different third wave, fourth wave that we are talking about. So initially, first variant has been isolated from UK and in September 2020. So now WHO is giving specific names to it. So before it was given B1.1.7, but now it is called as alpha variant. Then South African variant has been isolated, which is called as beta variant. And then the Brazilian variant, which has been identified in Jan 2021, which is called as gamma variant. Now the Indian variant has been named it as Delta variant. That is what people are thinking, that it is all spreading to different countries because we don't have complete ban on international travel. And so variants are very, very important, as I told you just now, that with respect to its transmission as well as virulence. And so to achieve herd immunity, this is going to be a problem. If the virus is too much changed, then the vaccine may not be able to protect it. So apart from that, there are some other a glimpse of some of the variants also in different areas of globe. So people are continuously monitoring. This is all sequencing information with respect to the isolates which are collected from different states of our country, and people are monitoring the variants which are coming up, and then they're seeing it, how it has been spreading. Okay, so this is the sequencing information that has been highlighted here. So now we get into, after knowing all the details with respect to the virus structure, genome organization, pathogenesis, symptoms, symptomatology, all that, once we have an idea, we need to get into the real picture where we can control this through the detection and testing, and then using the vaccination as well as therapeutics. Okay, so the detection is very straightforward. Actually, we can detect the virus in the nasopharyngeal swabs in general. So people try to isolate the RNA and do the RT-PCR because it is RNA virus. You have to do the reverse transcription PCR, and people are using real-time PCR so that you can know the amount of the viral load also, like quantitation of the RNA also you can do it. And then if at all if you are going for serological tests, then you have to wait for some time because you need to see the antibodies in the serological test. So for a viral antigen, people are trying different, they're coming up with different other tests also, which are in the laboratory right now, which are not into the commercial place. And then the antibody tests can be ELISA and LFA. LFA is a quick test, where a point of care test where you can use it, then you get an idea of about antibodies which are present in the body. Okay, so this is when to do the PCR, RT-PCR. It has been shown here, like you can use RT-PCR after 1 week of it. You can get an idea. You can get the PCR positive immediately. You may not be able to get positive reaction. That's how people are getting into problem. So they think that they're negative, but the virus will be multiplying and it will be entering into the next stage. So one has to be careful, and then you know that once the seroconversion takes place, convalescent time, you get the antibodies, and that's where you get the antibody test positive. So you need to know when to do this test. That is also important because some of the people are cheating. and doing the antibody tests at the first week itself, where you don't see any antibodies at that particular time, and unnecessarily these old people and people who are not having proper idea about these things, they are paying money to the centers, thousands of rupees, they are losing it. Okay, so detection and testing, as I told you, these are all the fluids we can take it, but blood and urine you cannot take. Nasal swabs, generally 2 to 3 days. You need to have at least BSL-2 facility, and it is most accurate. RT-PCR and mRNA can be detected anytime. You can do it once we infected. Blood, generally you have to go for antibodies, both IgM and IgG detected, but you need to wait for 14 days for that. So how do you identify that people got infection? You can look at for symptoms. So, or you can do a PCR test and then serological tests also, like that's what it has been given here. So RT-PCR, how we can do RT-PCR here, given a flow chart, but I think this is for students only, don't worry. And this is an ELISA, again for students I've given, and this is LFIA, so lateral flow immunoassay, where if at all, if you have COVID positive, you will get 2 lines. If it is COVID negative, you get only 1 line. Based on the line point of care test, there and then and there you can identify whether the person is infected by COVID or not, I mean SARS coronavirus or not. So apart from that, chest CT scan also very important. So the lower lobe, especially you have opaqueness at the lower lobe. Based on that, you can detect the thing. And apart from that, there are some blood markers and enzyme markers. You can identify them. So people are taking a decision based on that also. So prognosis, so as you know that the critical patients has to go for hospitalization, and they need to have the ventilator support. It's very, very important. And then the markers I've given here, hepatic markers, cardiac markers, and renal markers. Based on this also, you will be able to identify the patient's severity and the complications with respect to the patient. You can monitor the biochemical markers and find out, and that will help for the patient recovery. For the treatment regime, it will be of much help, actually. So that is what it is given here. All these prognostic markers can be checked. Increase or decrease can be really looked at it. Okay, so here again the D-dimers with respect to the blood clotting. So whenever there is a issue with respect to the blood clotting, then the D-dimer will be increasing. You can identify that with respect to it. So diagnostic summary, if you look at it, so clinical features are very important. Laboratory tests can be done. Imaging chest CT scan will be of use. Molecular examination by real-time PCR and other types of RT-PCRs. Immunological, you can do it with antibody tests. And novel techniques can be, lot of techniques are coming up, but still in the laboratory session. So once you get into the prevention and control of the disease, so many things are coming into the picture. Like you need to maintain the PPE isolation. There are many other testings and environmental measures. All together will come to the reduction in transmission. It is very, very important to reduce the transmission to get into original control measures. So medical treatment, as I said, there is no specific treatment actually. Only supportive care is there. Fluid therapy, oxygen support, and vital organs. And then mechanical ventilation, of course, for the acute respiratory distress syndrome patients you need to have. So now we are getting into the vaccines. So actually, to develop any vaccine, it will take a long time. That is what this picture is showing that. So to go into the vaccine manufacturing to the higher scale, you need to go through all these processes where you need to grow the virus and you need to make sure that it is prepared so much, and you need to do the clinical trials, and it has to go through the regulatory authorities, and then it has go to the large scale manufacture. So if at all, if you want to develop a new vaccine, it has to have all these characteristics with high quality, high stability, high efficiency, high safety, cost effective it should be, and it should be amenable for large scale production. It should be of high yield, and it should be of easy handling. That is the thing that we want right now, but we don't have so much time. So one has to go for the repurposed vaccines. So people are trying to use different strategies which are already tried for different viruses, and these are all the different types of vaccines which are people are trying onto it. So conventional vaccines like live attenuated and inactivated vaccines, and modern vaccines like recombinant DNA technology based vaccines like vectored vaccines, and then recombinant subunit vaccines, VLP vaccines, and DNA vaccines, and RNA vaccines, of course, which are all in place. And then people are trying globally for development of all these strategies. And then apart from that, you have monoclonal antibody therapy, and then therapeutics are in place, and stem cell therapy also people are trying, and then herbal treatments, especially Chinese, some of the herbal treatments, like, recently Anandaya Mandu has become very popular. We don't know the real facts, but it is also a belief that people are getting recovery with respect to it. So most probably the combination therapies may work, and we may have to go for novel vaccines, and nano formulations, of course, is going to be a boon in the future. So this, I've given a glimpse of different types of vaccines which are in trials, like a Moderna vaccine, where you will try to get the mRNA of S protein based on mostly S protein only. Even the DNA vaccine also S protein, and then the adenovector vaccine also, where the adenovector will become, they'll make sure that adenovector is replication competent, but it is not virulent also, but it will be able to produce a lot of S protein. And then people are trying to use different strategies altogether, and that is what I've given a glimpse of it. Okay, so a list of vaccines, so which are in trials, and which are the highlighted ones are the Moderna vaccine is up, and then we have Bharat Biotech vaccine, and Serum Institute of vaccine also is up, and the Pfizer vaccine is also up, and we have all these in the place. Like we have 2 vaccines in US, and we have Sputnik V in Russia, and we have Covaxin in our country. It is completely indigenous vaccine which has been developed by ICMR, NIV, and Bharat Biotech. 3 together, they come together and develop the inactivated virus vaccine, which should be an effective vaccine actually. And then Serum Institute of India got the technology, and then they are developing the adenoviral based vaccine again to get based on the S protein. So that is about the vaccines pretty much. And then we have therapeutics. Many therapeutics, as Vijay Prasad said that they're all repurposed again. They're all not directly working on virus, but some of them which are working for HCV and SARS coronavirus. Whatever people they have tried for it, they're trying here. And how they're trying to stop the thing is that they are trying to use some of the therapeutics which can stop the entry, fusion, and assembly of the virus by putting the inhibitors into it, or protease inhibitors. You know that TMPSS is important. If you use the protease, then the entry can be stopped, and RDRP inhibitors. So the RNA-dependent RNA polymerase, if it is inhibited, then multiplication may not take place. So this is antibody therapy, where people are trying to use the monoclonal antibody therapy, where it can bind to the ACE2 receptor, so that the coronavirus will not be able to enter into the ACE2 because of the binding. So these are all the list of different therapeutic molecules which are in use, and then the monoclonal antibodies also that are in use. So again, the green color ones are already tried ones for other viruses. People are using the other one, the blue ones also, based on the situation of the patient. So this is what the immune response pro-inflammation can be reduced by using all these different therapeutic molecules. That is what people are trying for. So when they're using all these therapeutics, they're trying to reduce this inflammation, and then they're trying to recover the patient and save the patient from the death. So that is what is happening. So stem cell therapy has been come up recently. So they're telling that the stem cells can take care of the immunoregulation, and they can reduce the effect of these inflammatory cytokines, so that the patient will be recovered and he'll become normal. So stem cells are doing a great job in immunomodulatory functions. Okay, so that is what they're claiming. And there are many challenges, lot of challenges are there. as we know that the diagnostic test we need still the point of care tests has to become into picture, because we have a real-time PCR authentic test, but it takes 1 to 2 days. So we need to have some tests which can be much more quicker than that, and cheaper tests also needed. Real-time PCR is costly, and then highly mutable single-stranded RNA genome is a problem, because it is getting into mutations, and then complete biology and strain information is not known completely. We need further investigation on that. Long-term viability is again problem onto the surfaces. So that is how it is getting into the more transmission, and recurrence is again problem. People who are recovered from the infection, as per the immune mechanism, they should not get infection, but there are reports that the people are getting again secondary infection, second time infection. So that is going to be a dangerous thing. Fecal-oral transmission has been also reported, and stool samples are we have found the RNA, SARS coronavirus RNA in the stool samples, and that is the way that it is an indication that it is into the GI tract. That is also a problem. And temperature effect and other environmental factor effects we don't know on the virus, so that has to be studied very well. And there are right now 2 coronavirus vaccines are present, but we don't have still large scale manufacturing facility. That is again problem. We are still getting a lot of vaccine scarcity, and that is not good actually in a big country like India. So if you have only less vaccines, it is not going to help much. So again, the capacities has to be built at a faster rate, which is very tedious and money and time consuming. Large clinical data is also has to be up, and it has to be conducted, and the established cell cultures and laboratory animals has to be tested, used for this testing, and additional manufacturing steps and formal toxicology testing has to be done, and complete with all the safety and efficacy trials with all the new products, new vaccines which are coming up. So how we can reduce this plateau, I mean, how we can make it as a straight line? We have to use this guidelines like SMS we have to use, like all these things I don't have to go through. So on together, totally what you can do is that you, as sir said rightly, you should not get panic. You need to be having the strong willpower and hope, and the balanced and simple food, and stress has to be minimized. Good sleep helps, and alcohol has to be avoided, and smoking, of course, it has to be avoided. And then social distancing, good hygiene, and mask. And then the stock up of food supplies are important. So masks, again, N95 is the best, but homemade mask, if at all, if you're using it, is better to have 3 to 4 layers of masks. And decontamination, you can use all these things. And surface, if at all, if you use for 3 to 5 minutes, everything has to go and virus should get inactivated. So what is the conclusion that we can get up to all this information is that social distancing is very, very important. We need to stay home unless otherwise it is required. We should not go out, and so that we can stop the spread, we can save the lives. So vaccination, at least, of course, it has to be increased, and vaccination should work, and it should be the ultimate criteria that one has to take so that you can end the pandemic. Antivirals also helps, and herd immunity is a kind of question mark and very risky. We cannot wait till we get the herd immunity, and then revaccination cases or reports are there, but only few cases are there. So there are again the conclusion that we can take is that the innovative funding is required. Government has to pour money for large-scale development of vaccines. Novel adjuvants and delivery methods can be tried so that you can get much more efficiency with respect to vaccines. And right now we have 2 vaccines, but more vaccines has to be get into the picture, like partnerships and R&D funding to fulfill the knowledge gaps with respect to biology and variants has to be done. And then we need to check the variants for their vaccine efficacy and safety. And then we have good large manufacturing facilities in India. So many tech transfer models can be taken up so that we can take the technology from other countries and we can develop it and we can make money out of it. So that is one idea. And then we can stockpile the coronavirus vaccines so that the viruses, if at all, if they are going to come as third wave, fourth wave. So we will be able to get ready, and then we can tackle the situation. So all the information with respect to all the slides I've taken from PubMed mostly, and then some of them are Wikipedia, and I've taken permission from Professor Sambareddy, USA, from his presentation, and then all the other authentic sites are from WHO, FDA, and Our World in Data, and CDC. With their permission, I've taken it. And then this is our department, Department of Virology in Venkateshwara University. This is unique with respect to offering MSc PhD in virology. So this is the only university which is offering MSc PhD in virology across the country. And I just want to leave all of you with a caption that we need to be very careful, because the viruses will be keep coming. If not SARS coronavirus, tomorrow Ebola might come, or any other virus may come, and we may get into again problem. So one has to be very careful and has the awareness with respect to the virus biology and the transmissions. Okay, I would like to thank you, all the participants for their patient listening, and I would like to thank the organizers and administrators for this wonderful opportunity. I would like to answer if at all, if any questions are there. Thank you so much.
Thank you so much, Professor Hema, Madam. Thank you so much for your detailed and scintillating talk. It was very elaborative. We enjoyed a lot. I think it's a time for the discussion or any queries from the students or any other participants. If there are no queries, then we can move to the next speaker.
Can I ask a question?
Yes,
Yeah,
yes.
yeah, please.
Please.
I'm Dr.
Please
Keshav,
unmute
actually.
and then talk.
Yeah. I'm already unmuted.
Okay.
Can you all hear me?
Yes.
Yes.
Actually, I have posted a question in the question and answer thing, but because I have like
Okay, you
facility
can
to
ask. No problem.
yeah, I can. This question is regarding like variants, and actually, Madam was showing different variants with numbers and alphabets. So, what those alphabets and numbers they represent?
Actually, numbers. See, they have taken a criteria initially to give the nomenclature of the variants, like the place of the thing, and they have taken how many, like they are sequencing number of samples from different areas, right? So initially they were having a type of nomenclature, but WHO now they have changed that one, and they are naming it as alpha, delta, gamma variant. So you don't have to worry about those numbers, okay?
Yeah.
They're
Now,
all
actually,
like,
earlier
they're representing the sample number and the place of the thing, and they have a certain criteria for classification. I mean, nomenclature. I really don't have any idea about that. I'm sorry.
Okay. Yeah. So there are I think different mutants, right?
Yeah, the numbers which are representing their different mutants. Mutants means they have certain significant amount of changes in their structure. I mean, in their sequence.
Yeah. So they are all actually sample, like from the first sample they have collected from a patient, right? The number represents for the patient identity.
Yes.
Okay. And one more question, like because this is therapeutics, you have discussed I think many therapeutics, but one thing I think maybe you have missed, because this one is from our own nation, 2-deoxy-D. I think you have missed that in your talk. Am I right?
Yeah. Probably I would have missed it, but actually those are all the recent data that has added, and there are many other molecules also has been added up. But concurrent scientific journals, whatever they have given the list, that only I have taken.
What is the efficacy of this one, true deoxygen? Do you have any idea about it?
Actually, it is having a particular mechanism, but exactly I don't remember right now. Okay.
Okay. Okay. Thank you.
Yeah, no problem.
Any other participants, if you have questions, please raise. Anybody has any questions?
TCS.
Yes.
Good morning, sir. Please.
Unmute TCS.
TCS, sir, could you please
Yeah,
unmute?
yeah.
Thank you.
Thank you for giving the opportunity, madam. Professor Hema, madam, thank you for your presentation. But this coronavirus is a virus. A group of it's a one of the group of coronaviruses. Is there any evidence that other genus like corona? It's a group of viruses, right? Any other coronavirus is presence. No, what I'm thinking is this is they are telling them this strain and that strain. Is there any other genus in that corona virus group? Any other genus telling about the strains and species only? Any other genus is mixing with this corona virus?
No, sir. No, sir. Right now, no, sir. Right now, there is no evidence that other genera viruses are mixing with this. There is no evidence for that. Actually, it is very straightforward, sir. Like variants are coming up because of the errors done by the RDRP. So it is not like influenza virus where you have a segmented genome, and when it is going to the other animal or wherever it is going, where it will be mixed with 2-3 fragments of the genome, segmented genome. It is not that sort actually. It is RDRP is doing the replication, and while doing it, it is tend to make the errors because it is RNA genome tend to undergo mutations because of the errors made by the RDRP.
But after seeing the after seeing the symptoms, what I suspect is a lot of coronaviruses, including different genus also. That is my suspect only. It's invading the whole globally. It is invading the group of coronavirus. Do you agree with that?
No, sir. No, sir. There is no, because it is a single copy, sir. It is single copy and RDRP is binding and making the RNA genome. Right. So where is the mixing will take place? If at all, if it is segmented genome, then they
No, that is my suspect only. That is, I'm not telling. I didn't go through any literature also, but is my suspect is it's a coronavirus. It contains, this group contains a lot of viruses. No, we are suspecting only genome by taking the genome content and we are doing RT-PCR and all. That's okay. But what my suspect is other corona group, other genus in the corona group also is invading the globe globally. That's what I'm suspecting. Do you agree with me or not?
Other genuses of coronaviruses are spreading. That may be true, okay. But in the virus, I mean, in the variant, there is no way that the 2-3 genera of the groups, I mean, the viruses will come and make one copy. That is not going to happen.
But we are talking about only strains only, not even species level. But how about the genus level? That's what. No, it's just a general question.
That's what, sir.
We are telling.
With respect to the variant, so it is about the single RNA genome that we are talking about. So there may be spread of other coronaviruses also, which has to be sequenced and worked out and identified. But with respect to the multiplication, it is not that 2-3 viruses sequences are going to mix up and make a different copy altogether. It is not going to be the case.
Okay, we are seeing a different symptoms. I don't think so. The symptom is, it varies like anything. Somebody will get. Even I heard that one of the article that one of the child at almost like 23 years old, that boy got, infected in the brain also. There they found the coronavirus also. So a lot of symptoms. There is no guarantee these symptoms also. We are telling about 15 or 16 symptoms like high fever, cough, dry cough, kind of things. So this much symptoms are there with single virus.
Sir, it could be, it is not just because of the virus, no, sir. Virus is creating some other complications, right? During the pathogenesis. So, and we cannot exactly say that it is just because of the virus infection, but the virus infection can try to trigger the other things also, depending on the person's age and comorbidities and sex and many other things. With respect to the genetic factors, the person's genetic factors, many other things will work on it. So we cannot say that, but virus infection can trigger many other aspects.
In our last sentence, I guess I remember, there is a chance of Ebola, but Ebola is almost eradicated 2-3 decades back in Congo and other African countries, right?
Sir, I'm not telling that it is going to come. I said that there may be a chance that many other diseases may come. You see, once upon a time, SARS coronavirus 1 also eradicated. I mean, it is not there. It has come, right? So like that,
yes,
there
in
may
Taiwan
be
and
chance.
all other places.
That's what I said. I just want to give a significant concluding line. That's all.
Yeah, thank you so much.
You're most welcome, sir.
Any other questions from the participants?
Thank you, TCS sir, for your queries. Hope you
have to say Dr. Kesav also, madam.
Yeah.
You know me, that's why you are telling thanks to me, but you need to say thanks to Dr. Kesav also. I don't know who is Kesav.
Yeah.
If anybody has any questions, if no questions, can I ask one question, Hema?
Yes.
I would like to ask one question. So what are the vaccine development strategies to cope up with the upcoming variants? Is any research on that?
Absolutely no idea on that. One has to deal with it. That's all we can do. So actually, as such, the whole inactivated vaccine should take care of everything because it has covered most of the epitopes, all epitopes, in fact. But the efficiency with respect to the whole inactivated vaccines, again, it has got its own problems because it is not going to multiply.
Exactly.
So S protein is going to cover only part of the neutralizing epitopes. So how far it is going to work with respect to the upcoming variants, depending on the variations, only we can decide. I mean, one has to work and then only we can tell. As per my idea, I think the whole inactivated vaccine should take care of.
Okay. Any mutations in the virus also can take care of by the inactivated virus?
That again, one has to go for vaccine preparation, but again, you never know, right? How it is. It has to work out again in the clinical trials. So then only we can say that.
Okay. Yeah. Okay. Thank you. Thank you. And if no questions from the participants, I would like to thank Professor Hema for a very informative and wonderful speech regarding the COVID-19 therapeutics and vaccines. Thank you very much, Hema.
You're welcome. I once again thank the organizers for the opportunity. Okay. Thank you.
Thank you, madam.
Thank you, madam. Thank you.
Now, coming to the next speaker of the webinar, Dr. Pratap Bingi. Dr. Pratap Bingi did his MBBS from Kurnool Medical College, Kurnool in 2013 and finished his MD, Internal Medicine from Kurnool Medical College, Kurnool in 2016. And he secured top marks in the university post-graduation examinations and achieved University Gold Medal. And he has been awarded with the Dr. Pinakapani Gold Medal for the Best Outgoing Postgraduate for the year 2016. And he has also awarded with the prestigious Amicon Gold Medal at national level and also awarded with the Commendation Certificate for the Best Postgraduate in 2016. And he is a member of IMA India and AP India. To his credentials, Dr. Pratap has a certificate in diabetology from International Diabetes Federation, IDF, EACS, and also certificate in advances in management of thyroid diseases from American Thyroid Association, and also certificate in primary care cardiology. Coming to the work history of Dr. Pratap is currently working as assistant professor in Government General Hospital, Kadapa since 2017. And he has good experience in the treatment of patients in OPD medical ICU and cardiology departments, training of postgraduates in internal medicine and nursing students and paramedical students, and also in charge of the medical ICU department. And Dr. Pratap is a consultant physician in the Himalaya Multi-Speciality Hospital at Kadapa. And Dr. Pratap has also one YouTube channel. So now I welcome Dr. Pratap for his deliberate in this COVID pandemic. Thank you, Dr. Pratap, for being with us today. Over to Dr. Pratap, please.
Thank you, madam. Thank you for the kind words about me. First of all, I'd like to appreciate the previous speaker for extensively covering almost every topic in COVID-19. And she has left me with very few things to discuss. The madam has covered almost everything, but in this session, I'll be discussing more of management of COVID-19 rather than about virus structure and all. So it will be dealing with the clinics, like more clinical management, how to diagnose the case of COVID-19, what are the symptoms, and how to categorize the patients into different categories, and how to treat according to that. So now, so this is today my topic for discussion is about management of COVID-19. So as already said, coronavirus is a novel disease. So it was COVID-19 is an infectious disease which was caused by novel coronavirus, which was recently discovered. The structure and all already madam has said. The incubation period of COVID-19 is thought to be it was supposed to be around 14 days, and it may start anywhere between 4 to 5 days from the day of symptom onset. So the incubation period ranges from 4 to 5 days to 14 days. COVID-19 affects different people in different ways. So there is a wide variety of symptomatology, and if you see out of 100 patients who are infected, only a few patients, like more than 80% of the patients, they will recover with a mild disease, and they will have a very, very mild symptomatology. And usually they will recover from itself. So only a fraction of people, like 20% of the patients, will develop moderate disease, and only a few out of them, like 1 or 2% out of them, will succumb to serious illness, and that will be life-threatening. So in fact, like other normal viral infections, COVID-19 is a self-limiting disease in more than 80% of the patients. It doesn't need any treatment, so it will be self-limiting, like symptoms will be very mild in more than 80% of the individuals. So usually 80%, most of the people will develop mild to moderate disease and recover without any hospitalization. So the most common symptoms that are seen in patients with COVID-19 is fever, a very important symptom almost seen in more than 90% of the individuals. So fever usually lasts for a day or two. So it is not like the fever will be there continuously. So fever will usually be lasting for only a day or two. So then the patient will become completely symptom free. So then the patient will develop other symptoms like tiredness, which is extreme fatiguability. So most of all the patients, like more than 90% of the patients who develop coronavirus infection, they will be having this extreme fatiguability. So that is one of the important symptoms patient will say to you. So extreme tiredness, extreme fatiguability is the marker for COVID-19 infection. So when a patient has got a fever of 1 day or 2 days duration with extreme tiredness, always suspect that he may have a COVID-19 infection. So the other symptoms are dry cough and breathlessness. So this is the range of symptoms. Patient may be having cough, fever, headache. So which is one of the worrisome symptoms. Extreme tiredness, may be having chills associated with fever, sweating associated with fever, generalized body pains, back pain, difficulty in breathing in the form of SOB or dyspnea. So it is not must that every patient should develop all these symptoms. Usually, most of the patients, like more than 80% of the patients, will have a very few symptoms. Like he may be having a fever of 1 day duration, or the patient may be having only an extreme fatigability for a week or 5 days or 1 week, and then it will be self-limiting. So these symptoms are not universal, like this should be present in all the individuals. It depends. So most of the patients will be having a few symptomatology. Only a fraction of patients, like 10 to 20% of patients, will have respiratory difficulties, like respiratory symptoms, symptoms of lung involvement, like cough, which is dry in nature without any production of sputum, and breathlessness, so which is usually exertion. So exertion breathlessness means when the patient is walking or doing some sort of activity, then only he will develop breathlessness. That is exertional breathlessness. So the less common symptoms are muscle pains, body aches, sore throat, diarrhea. Like usually the patient may be complaining of diarrhea in day 1 or day 2 of illness, conjunctivitis, headache, loss of taste or loss of smell. So this happens to be one of the specific symptoms of coronavirus infection. So this is not a universal symptom, but it is seen in almost 50% of the individuals. So the loss of taste or smell points more likely towards coronavirus infection. Some patient is having the loss of taste or smell, so most likely it will be a COVID-19 infection. So some people may be having a rash on the skin as what we see in other viral infections. So or there may be a discoloration of finger or toes. So these are all the less common symptoms. Almost this will be seen in a few patients, like it will range from 20 to 50% of the patients. But fever and extreme fatiguability is almost universal. Fever and extreme fatiguability is seen in almost 90% of the patients. So keeping these symptomatologies into consideration, so when you are seeing patients who are having symptoms of fever or fatiguability in the background of all these or some of these symptoms, then we will suspect a COVID-19 infection. So once we suspect a COVID-19 infection, then the testing strategies are different. It depends on how many days the patient has got symptoms. The patient is having symptoms which are ranging for less than a week. So when the patient is coming to you within a week, so in that case, when the patient is having symptoms which are less than a week, in that case, when we test the patient for either an antigen test or an RT-PCR test, then the patient may yield positive. So usually the antigen test or the RT-PCR test should be performed preferably within 1 week of illness. So then only it will be positive. Else, if it crosses more than 7 days, there will be false negative RT-PCR or COVID-19 antigen test. So the testing time is very important. So for that, we need to, so the testing time is very important. We need to take a clear history from the patient to know the duration of symptoms. When the duration of symptoms are less than a week, so preferably perform microbiological tests like RT-PCR, or we can go for a rapid antigen test. If the symptoms range more than a week, then performing RT-PCR or rapid antigen test is not must, and it will not yield a positive report. So in such cases, we will go for an advanced test like CT scan of chest, which will yield lung involvement, which will show you the extent of lung involvement in that case. So performing a CT is better if we do it after 5 days of illness. So this is a very, very important thing that should be taken into consideration while treating the patients of COVID-19. So then there are a few blood tests, so which is like which shows the inflammation that has been happened because of this COVID-19. So the inflammatory phase, so which is because of actual inflammation, the lung is damaged and other systems are involved. So that inflammatory test like DNM, CRP, ferritin, LDH, and so on. So those tests we are preferably performed after 1 week of illness, as the inflammatory phase usually starts from day 7 to day 12. So in such period, if you do inflammatory test, then that will help you in treating the patients. So this is about the symptomatology. So taking these symptoms into consideration, when once we see a patient, so in such patients, we need to like test them, or we need to examine them to categorize them into different severities. So there are three groups of severities, like mild group, moderate disease, and severe disease. How to categorize them into mild, moderate, and severe will be seen. So the mild disease is what the symptoms are very limited, or the symptoms are limited to only upper respiratory tract. So upper respiratory tract means when the symptoms are limited only to the nose, pharynx, mouth, or up to the level of larynx. So which is not involving the lungs. So when there is only an upper respiratory tract involvement, like patient may be having a dry cough, may be having a sore throat, like there is a sore throat, may be having a dry cough, may be having a sore throat, or patient may be having fever without breathlessness. Without breathlessness, a very, very important thing. Patient don't have a breathlessness. If the patient is having a breathlessness, then that signifies there is a lung involvement. So if the patient is having no breathlessness and he has got only few symptoms like fever, sore throat, and only dry cough, then consider this as a mild disease. In such patients, checking of oxygen saturation. So a very important thing which we do with the pulse oximeter. So the normal oxygen saturation is more than 96 in an individual, in a normal individual. It will be usually up to 98 or 99. So when that saturation levels are normal, like it is more than 96, without any breathlessness, and the patient has got mild symptoms like fever, cough, or sore throat, or body aches, or headache, without any breathlessness, then you consider this as a mild disease. So in cases of mild disease, most of the patients, that is up to 80% of the individuals, will have this mild disease. So mild disease is very easy to treat. So most important thing is to categorize the patients to know whether it is mild, moderate, or severe. So if you are confident that this patient has got a mild disease, then it is very easy for you to treat. So then the treatment guidelines for that patient is home isolation and home care. A very, very important thing. So most of the patients can be maintained under home isolation, but there are many things what the patient should be educated before sending him to home isolation or home care. So he need to know about what he should do at home, what he should eat at home, when he should come to a doctor, what are his warning signs, when he should take a medical attention. So he should know everything, what he should do and he should not do. Do's and don'ts should be known, or it should be taught to the patient before sending him home. So the do's that should be done in home isolation are the patient should maintain a physical distancing. So, or preferably, he should be kept in a separate room. So, he should be kept in a separate room where the lavatory or the toilet is separate for that individual. So, he should be kept in a separate room. Or if not possible, he should maintain the physical distancing of at least 2 meters from other individuals. Then is mask use inside home. So when the patient is positive, when he is sent for home isolation, he should be strictly advised of using mask in the home also. So indoor mask usage. So indoor mask usage and strict hand hygiene. So repeated hand washing. It should be done. Then give him some pills for symptomatic management. Like when there is a fever, give some antibiotics. When there is a cough, give some antitussives. So give multivitamins which boosts his immunity, and always advise patient to take plenty of water, as hydration decreases all the symptoms. It decreases the fatigability. It improves the patient overall general condition. So he should be staying in contact with the treating physician, whoever may be the doctor. He should be staying in contact, and at home he should be given a chart to monitor his temperature, his oxygen saturation. So both the temperature and the oxygen saturation should be monitored, and it should be filled every 6 to 8 hours, and that chart should be brought to the patient, to the doctor in the next visit. So then when he needs a medical attention, when he should take a medical attention. So he should take a medical attention immediately if the patient has got a symptom of breathlessness or difficulty in breathing. So the difficulty in breathing implies the patient has got a lung involvement and that has moved his staging in severity from mild to moderate disease. So once he is having a moderate disease, he develops breathlessness. Or when the patient is having severe cough, which is not coming down with the routine antitussives or the treatment what is given at home, or when the patient is having high grade fevers which are not coming down with the routine antipyretics, or if the symptoms are lasting for more than 5 days, a very, very important thing. So if the symptoms are lasting for more than 5 days, or when the patient has got respiratory symptoms like severe cough, severe fever or high grade fever, or when there is a difficulty in breathing, these are the symptoms which are considered as high risk symptoms. And once the patient develops this sort of high risk symptoms, the patient should be immediately going to a doctor and he should get admitted. So this is about the mind disease. So this is the what we should like. What are the do's and don'ts that should be educated to the patient? And what are the medications we will give to this patient who is there in the like mild disease group? So as I already said, multivitamins, that is vitamin C, zinc, and vitamin D should be given. Along with that, the patient should be given some antibiotics and antivirals depending upon the risk stratification of the patient. If the patient is belonging to high risk groups. So what are high risk groups is when the patient has got some comorbid illnesses, like if the patient is having a diabetes, or if the patient is having hypertension, or if he is old age person, or if the patient is having some chronic diseases like heart disease, kidney disease, liver disease, all these things are considered as a patient having a high risk profile. For that patient should be given certain antibiotics like ivermectin at a dose of 200 micrograms per kg once a day for 3 to 5 days. Second one is doxycycline at a dose of 100 mg per day, 100 mg divided in like 100 mg twice a day for 5 days. So these two drugs can be given, and we also give certain antibiotics like azithromycin or certain antivirals like oseltamivir or liver antivirals like favipiravir. So it depends on whether the patient is having symptoms, or so the things that should be taken into consideration is whether the patient belongs to high risk group or the patient is having more symptoms. So in such group, we will give antivirals. So antivirals like favipiravir or oseltamivir can be given. And when the patient has got symptoms of respiratory onset like cough, or if they have mild breathlessness, in such cases we can use inhalation steroids. Inhalation steroids like inhalation budesonide, it should be given at a dose of 800 micrograms twice a day for 5 days. So it should be given at 800 micrograms twice a day for 5 days. So usually this will bring down the symptoms of cough and breathlessness or respiratory symptoms. So most of the patients of COVID-19 belong to this category. Most of the patients will improve with this simple treatment what we give at home. So only a minority of patients, like about 20% or 15 to 20% of the patients, will have a moderate or severe disease. So now let us move on to the next thing, like moderate disease, the second group. So moderate disease is when the patient has got all the symptoms what we have discussed in the previous section, like patient having fever, cough, breathlessness, fatigability, or the patient is having headache, body pains. So all those are some of those symptoms. In addition to that, when there is a lower respiratory tract involvement, that patient is having a breathlessness and the respiratory rate is more than 24 per minute. Very, very important one. The respiratory rate is more than 24 per minute. In such cases, when the respiratory rate is more than 24 per minute, or when there is a breathlessness, second point is when the patient oxygen saturation. So what we already said is normal is more than 96. If the patient oxygen saturation is 90 to 93 or 94, so then that patient is considered as having a moderate disease. So when the saturation level drops below 95 and it is above 90, so when it is dropped below 95 or it is above 90, so then that patient is considered to be having a moderate disease. In such cases, these patients should not be sent home. They need to be admitted in medical wards, and if needed, when the saturation is less than 94, they need to be given with oxygen therapy. So the central mechanism or the central thing in treatment of patients with moderate and severe disease is oxygen. So the only effective, so there are only few effective therapies for COVID-19 until now. Out of those few effective therapies, the most important and the first ranked therapy is oxygen. So oxygen is the only proven therapy which will help the patients with COVID-19. So the oxygen therapy that can be given with different types of masks, like we can give it normal mask, or we can give it non-rebreathing mask, or if the severity is more, in such cases we can give it mechanical ventilators, either in the non-invasive way or invasive way. That we'll be dealing with in the next sections. So now when the patient is having a moderate disease, so it is always advisable for him, for us to admit him in the ward and keep him in close monitoring with a treating doctor. A very, very important one. So oxygen support should be given in addition to the treatment what we are discussing for mild disease, like giving some antibiotics, inhalation steroids, antibiotics, multivitamins and all. The next thing what we need to do is giving an oxygen support. So with a target saturation of 92 to 96, a very, very important one. Target saturation, when we give oxygen, we can give at 1 liter, 2 liter, 3 liters. So depending upon the treatment patient severity, like saturation, so the target saturation should be maintained at 92 to 96 in normal individuals. In patient who has got a COPD, in patient who has got a COPD, like patient who has got a COPD, like chronic obstructive pulmonary disease, in such individuals, the target saturation should be kept at 88 to 92%. So the preferred device out of all the masks that are available, the preferred device for oxygenation is always the non-rebreathing face mask. This is the non-rebreathing face mask, where that is the preferred device. So here the patient, a mask is connected to a bag. So the exhaled air is going into this bag, and this is not inhaled again. So the non-rebreathing mask is one of the important treatment option. So it should be preferred for giving oxygen support in patient with COVID-19. Then prone positioning. So encourage the patient for prone positioning. So which is a very important aspect in treatment. So that itself, making the patient in prone position itself improves the oxygen saturation by 2 to 4%. So this is the guideline for prone positioning. So there are different positions which the patient should not lie in the same position every day. So he should lie in different positions for about 30 minutes to 2 hours. The first thing is the prone position, strict prone positioning like this, where the patient is lying in prone position with arms extended and legs extended, lying on the belly. So this should be maintained for 30 minutes to 2 hours. Next thing, the next 30 minutes to 2 hours, you should lie on the right side like this, with the right arm under your head. Then the third thing is again the patient is sitting up for 30 to 40 minutes, 30 to 30 minutes to 1 hour. Then he will lie down in the left position. Again come back to the first position. So this is how we change the positions regularly. For every 1 to 2 hours, the patient will be regularly changing, and the strict proning will increase the respiratory effort, and then the dead volume will be decreased, which increases the oxygen saturation by more than 2 to 4%. So very important thing which the patient should be educated. So lying down flat on the belly is the position of choice for treating a patient with COVID-19. Then, in addition to the therapies what we have discussed previously for mild diseases, In cases with moderate disease, there are two more options what should be taken into consideration. Number one is the steroids. So steroids is again the second effective treatment for COVID-19 and the proven treatment for COVID-19. So after oxygen, steroids are the thing which were proven, but steroids are always a double-edged sword. So we should take care of, like we should be limiting to the, or we should be strictly following the guidelines when we are prescribing steroids. So never prescribe steroids in a patient who has got a normal oxygen saturation or who has got no symptoms or who has got no respiratory symptoms. So that is in mild cases, don't go for oral or inhalation steroids, oral or intravenous steroids. Only inhalation steroids can be preferred if the symptoms are lasting for more than 5 days. So in cases with moderate disease, so it is always advised to go for anti-inflammatory or immunomodulatory therapy, that is steroids, most preferably in injectable way. The preferred steroid of choice is methylprednisolone, or we can go for an equivalent dose of dexamethasone. So these are the two preferred steroids for treating patients with COVID-19. So if you are going for a methylprednisolone injection, give at a dose of 0.5 to 1 mg per kg body weight in 2 or 3 divided doses, usually for a duration of 5 to 10 days. So, or if methylprednisolone is not indicated or there is contraindication for methylprednisolone, then we can go for dexamethasone in an equivalent dose, so which is usually given for a duration of 5 to 10 days. So after 5 to 10 days, when the patient's symptoms are improving, then we can switch over to the oral steroid, preferably a methylprednisolone or a prednisolone. So then we need to taper the steroid and decrease the dose of the steroid and stop over a period of 4 to 7 days. So this is how we use steroids. Then anticoagulation. So the anticoagulation means there is a respiratory involvement and the patient is having a breathlessness, and if the patient is having some pulmonary infiltrates on CT chest, then that is an indication for giving anticoagulation. So the pulmonary infiltrates, what we see on CT imaging of chest, that is in the lungs, they will be like they will be lysed only when we do an anticoagulation. So the anticoagulation of choice is enoxaparin sodium. So there are newer anticoagulants now which are available. So we can give all the newer anticoagulants as per the doctor's preference and the patient's what the patient is having, like whether the patient is having a comorbid illnesses or not. So depending upon what comorbidities the patient is having, we will prefer the anticoagulation. But the basic anticoagulation is always enoxaparin sodium. So usually we do enoxaparin sodium. So that the thing what we should take into consideration before giving enoxaparin sodium is risk of bleeding, whether the patient is having any risk of bleeding. So if he is having bleeding manifestations, low platelet counts, or if there is some previous history of bleeding, so then we should not use enoxaparin sodium. Then considering, so then how to monitor these patients? So moderate illness. So now we have admitted the patient. When you have started on all the treatment. So now how to monitor these patients? So in monitoring, there are two ways of monitoring. One is clinical monitoring. The second one is biochemical monitoring. So on day of admission, so usually we will send the biochemical tests to the lab, like the basic blood routines like complete blood picture, renal function test, liver function test, and COVID blood tests. The blood tests which are specific for COVID inflammatory phase, like D-dimer, CRP, C-reactive protein, ferritin, LDH, interleukin 6 and all. So we will run a basic blood profile and see what is the baseline blood values of that patient. So after that, we'll admit the patient. We'll go for a clinical monitoring. So clinical monitoring is based on symptoms and signs. So first thing is assessing the symptoms of the patient. So in daily rounds or when you are monitoring the patient daily, so we need to assess the patient's severity of symptoms, whether the symptoms are improving or it is decreasing or they are static. Then is the signs. So examine the patient for saturation of the patient, whether it is decreasing or it is maintaining at a normal range, or whether it is improving. So monitor the saturation, monitor the respiratory rate. So the work of breathing should be monitored. So the respiratory rate should be monitored. So when the work of breathing, patient's work of breathing should be monitored and note the respiratory rate. So the respiratory rate, usually day by day it should come down. So if it is increasing, then the patient clinical severity is increasing. Then we need to note on what is the change in the oxygen requirement. Today, if the patient is on 2 liters of oxygen, tomorrow it is escalating to 6 liters of oxygen, and day after tomorrow it is to 9 liters of oxygen, then the patient is clinically deteriorating. So if it is 2 on day 1, 1 on day 2, and then no oxygen on day 3, then it is improving. So clinical monitoring is based on the symptoms and the signs. So the basic things what we should do is assess the symptoms, assess the oxygen requirement, assess the work of breathing with the help of respiratory rate, and assess the saturation levels of the patient. Serial chest X-ray or HRCT chest should be done only if there is a worsening in the patient or clinical status. So if the patient clinical status is worsening, then you need to see serial chest X-rays to know whether the lung involvement is increasing or not. Laboratory monitoring. So usually I said on the day 1 of admission, we'll do a baseline test, and then every 2 days to 3 days, once we'll do a complete blood profile. So to know whether their inflammation is increasing or it is decreasing. So then when to give remdesivir in these individuals? So the remdesivir, it is the antiviral drug. So whom we should give remdesivir is when the patient has got a moderate disease or a severe disease who is requiring supplemental oxygen, and then the patient should not have any kidney problem or a liver problem. So the renal disease and liver disease should not be there, and remdesivir should preferably be given within 10 days of onset of symptoms. So in that phase only, before 10 days only, we call the disease as a viral stage. So once the disease crosses 10 days, then is the inflammatory phase. The actual virus has already gone out of the body, but the inflammation that was done by the virus, that is causing the symptoms after 10 days. So that stage is called as inflammatory phase. In inflammatory phase, that is more than 10 days, no antiviral is required. There is no need of giving an antiviral drug. In inflammatory phase, only things that work are anti-inflammatory drugs, that is the steroids or the liver immunomodulator drugs. So usually remdesivir, if you are choosing to give remdesivir, that should be given within 10 days of onset. So the recommended dose is 200 milligrams IV on day 1, followed by 100 milligrams IV daily for next 4 days. So usually this is not to be used in patients who are not on oxygen support or in home settings, like when the patient is having a mild disease. So in mild disease, remdesivir usually should not be given. Then comes the third stage of the disease, that is severe disease. Severe disease is when a patient has got a respiratory involvement like breathlessness with respiratory rate of more than 30, or the oxygen saturation is less than 90% on room rate. If these two criteria or one of these criteria is met, then we call this as a disease of severe disease. So then this disease is called as severe disease. So in severe disease, this patient should be shifted immediately to an ICU setup, intensive care unit, and he should be given respiratory support either with a facial mask, that is a non-rebreathing mask, or if the patient is not able to maintain the saturations, oxygen saturation with the face mask or the non-rebreathing mask, then go for artificial ventilation with the help of ventilators. So either a non-invasive ventilation or an invasive ventilation. So non-invasive ventilation is keeping a face mask which is attached to a ventilator, and ventilator settings will be changed, and the ventilator delivers oxygen at a high flow. So that is non-invasive ventilation. Or the patient can be given HFNC, that is high flow nasal cannulas can be used. So usually this high flow nasal cannulas delivers 50 to 60 liters of oxygen per minute. So the only therapy that usually helps the patients in COVID-19 is oxygen. So for that, so we need to give as much of oxygen as possible, keeping into mind the SpO2 or the saturation should be maintained at around 90 to 93%. So intubation or invasive ventilation should be prioritized in patients who has got high workup breathing or high respiratory rate, where NIV or non-invasive ventilation is non-tolerated. Then use conventional ARDS protocol for ventilatory management. So what are these ARDS protocols? So usually ARDS is what is happening in COVID-19, where there is bilateral lung involvement and all the lung is white out. So in such cases, we need to go for a ventilatory management with the ARDS protocol. So again, the same thing like what we discussed in moderate disease, anti-inflammatory or immunomodulatory therapy and anticoagulation should be given with unfractionated or unfractionated heparin or low molecular weight heparin. And immunomodulatory therapy or anti-inflammatory therapy, methylprednisolone, the dose is 1 to 2 mg per kg body weight in 2 divided doses for a duration of 5 to 10 days. So both the anticoagulation and immunomodulatory and steroids should be given. Supporting measures as what we discussed, maintain euvolemia, that is the fluid status should be maintained, give adequate water. If the patient is in sepsis or the patient is in septic shock, go for higher antibiotics based on the local antibiotic profile. So when there is a septic shock, give fluids adequately and maintain or give inotropes. So monitoring of these patients, again, as what we discussed in moderate disease, clinical monitoring should be done and laboratory monitoring should be done. Clinical monitoring with the help of symptoms and signs like respiratory rate, workup breathing, SpO2 saturation monitoring, etc. And laboratory monitoring with the help of CRP, D-dimer, interleukin 6, so ferritin and LDH, which are considered to be the markers of inflammation. So serial chest X-ray and HRCT test should be done if there is a worsening in clinical condition. The same as what we discussed in moderate disease. Here, in severe disease, so after steroids, when the patient is not improving with the steroids, when the patient is not improving with methylprednisolone, so we can go for an immunomodulatory therapy with monoclonal antibodies. So like tocilizumab. So this is the monoclonal antibody which was used. This may be considered when all of the following criteria are met, or when there is a presence of severe disease, preferably within 24 to 48 hours of onset of severe disease. So when there is a significantly raised inflammatory markers, patient is not improving in spite of using steroids, when there are no active bacterial, fungal or tubercular infections, in such cases, we can give a trial of tocilizumab at a dose of 4 to 6 mg per kg in 100 ml over a period of 1 hour. So which may be helpful in patients with severe disease. Don't attempt tocilizumab in patients who are having moderate or mild disease. Then is the convalescent plasma therapy. Again, this is not very much useful, but it was being tried. Convalescent plasma therapy may be considered only when the following criteria are met. So this is usually given in early disease and moderate disease, not in severe disease. Preferably, it should be given in 7 days of symptom onset. No use after giving no use when you give after 7 days. So the most important limiting factor for this is availability. So availability of plasma therapy, plasma is the most important limiting thing. And this happens to be not very much beneficial, but it is being tried for moderate diseases. Then high risk patients for severe disease or mortality, this should be kept in mind. Patients who have got this sort of illnesses are at more risk of developing severe disease. They should be treated more aggressively and they should be monitored very frequently. So because they have got more liability of getting a severe disease. So we should keep into consideration these factors while we are categorizing the patients. Age of the patient more than 60 years, a patient is having a previous cardiac disease, hypertension, or coronary artery disease, a patient is having a diabetes mellitus, or other immunocompromised states like HIV, chronic lung disease, chronic kidney disease, chronic liver disease, or patients have neurological disease like stroke or obesity. So these people are considered to be high risk groups. In such individuals, treatment should be individualized. So it is not universal. We should individualize the treatment based on what disease the patient is having, and the monitoring should be done very frequently, and close monitoring is more important. So once there is a clinical improvement, discharging the patient is as per the revised criteria. So discharging the patient, when you see for a mild, moderate disease which are pre-symptomatic, is usually keep them in home isolation. So once the patient crosses 10 days of symptom onset, so then when there is no fever for 3 days or no symptoms for more than 3 days, so then this patient of mild disease is considered to be like he was cured from COVID-19, and before discharge also there is no need of getting repeat test done. Whereas for moderate disease, fever is usually resolved within 3 days and the oxygen saturation is maintained without support of oxygen, then you can discharge the patient after 10 days of symptoms onset. So when there is no symptoms like fever, breathlessness is not there, and there is no oxygen requirement, then the patient can be discharged. In moderate disease, the symptoms are not resolved and demand of oxygen therapy continues. So then discharge should be done only after resolution of clinical symptoms and the ability to maintain oxygen saturation at more than 94% for at least 3 consecutive days. For severe disease, discharge should be done when there is a clinical recovery, and usually testing should be done before discharging the patient in severe disease only. So in severe cases, testing should be done before discharging the patient. So RT-PCR testing should be done. So this is about the discharge policy. With this, we complete the topic. Now the session is open for discussion. If there are any questions.
Now I request the participants to interact with the speaker.
So if there are any questions.
ఎవరైనా raise చేస్తారు hands.
Okay. Okay. Then we can give please.
1 minute time. No one is.
Okay.
Can I ask you one question, Dr. Pratap? So why some of the patients, COVID-19 patients, are asymptomatic?
Hello?
Yes. Why some COVID-19 patients are asymptomatic?
It is not like asymptomatic. Once there is an invasion of a virus particle inside the body, the patient may be having at least 1 or 2 symptoms. So without any symptomatology, there is no actual infection that will happen. So at least there will be a few symptoms, and the threshold for that symptom differs in different individuals. For example, say if there is a body pain or a fever, so few people will take it as a big symptom, and some people will take it as a normal thing. Like see, symptoms are very wide here. For example, if there is a fatigue, most of the patients usually think of fatigue as a normal symptom because of excess work or something. They relate to lack of sleep, or it may be something like because of excess work and all, and they will not make it or bring it to the notice of a doctor. So it is not that they are completely asymptomatic. They'll have at least 1 symptom or 2 symptoms. They'll have at least a small symptomatology, but it usually will be like limited to 1 or 2 days. So it is not longing for loss, it is not long lasting. So usually they will not bring. So most of the people, adult population, in fact, most of the people will not bring mild symptoms which last for 1 or 2 days to the doctor. So that is how we miss the symptoms. Not that you don't have. If you throw into the details properly and ask him repeatedly, then he will say that he got a fever 1 week back, or he got a body pains 1 week back, or a cold 1 week back, a headache which lasted for 1 or 2 days. So usually those are the symptoms of COVID-19 are the common symptoms what we see in common cold. So that is why most of the people will neglect those symptoms and not bring them to the notice of the doctor.
Okay, thank you.
One more question, sir. So I would like to ask one question. Men are more prone to the COVID pandemic than women?
It's not that, madam. It is there is no rule that men are more prone, but the infection rate is more in men when compared to females, as the mobility of men is more than compared to females. So at least for India, so more number of males are coming out and more number of females are there inside the home. And as we know, the symptom, the infection is because of like non-containment. People who are coming out will be more prone actually than people who are there inside homes. So that is what. There is no like specificity like virus infection will be there only to men and it will be less to females.
Thank you, sir. Okay.
Anybody has any more questions?
Sir, good afternoon, sir.
Good afternoon.
Sir, if a person suffering from typhoid fever, sir, but he don't know, sir, he already suffering from sinus, sir. In that it is similar to COVID-19. Yes. No, sir. In that time, how can we identify COVID-19 symptoms, sir?
What is the common misdiagnosis that is happening in the community? So as most of the people think fever or body pains or fatigue and they attribute it to typhoid. So most of the people will be coming late in the disease and they are succumbing to death because of this problem. So see, for any patient, when we do a viral test, for any individual when we do a viral test, it usually shows positive. So even for a normal patient, normal individual also, if you do a viral test, which is the test for typhoid fever, it will show like it is positive in many, almost 90% or more than 90% of the patient, 90% of the normal. So usually the doctors who are not aware of COVID-19, what they'll do is they'll attribute it to typhoid fever and start treating with conventional antibiotics, and they'll not guide the patient properly. And these patients will think mostly like when the symptoms are increasing, that might be because of COVID, that might be because of typhoid, and they neglect and they don't think of COVID-19. So usually they come to a qualified doctor in a late course in the disease. So that is usually at 10 or 12 days where the inflammatory phase has already begun. So in such cases, when we treat also, the patient will not recover. So typhoid is one of the common misdiagnoses which is happening nowadays in the community. So how to differentiate real typhoid fever from COVID-19 is usually in typhoid fever, the patient will be having only fever without usually the typhoid fever, the patient will be having fever without any other symptoms like respiratory or upper respiratory tract symptoms like sore throat or cold or extreme fatigability. So whenever the patient is having fever with a cold, sore throat, and extreme fatigability. So extreme fatigability is almost universal in patients with COVID-19. So when there is an extreme fatigability, so always because we are there in an era of COVID-19, before testing for viral, test for COVID-19. So that is how we can easily differentiate. So before going for a test for viral or before going for a test for typhoid, it is always preferable to test for COVID-19, then go for other alternate diagnosis. Because now we should believe any fever is COVID-19 unless proved otherwise.
Okay, sir. Thank you, sir.
Okay.
So if there are no questions, now I would like to thank Dr. Pratap for enlightening us regarding the COVID-19 management. Thank you very much.
Thank you, sir. Yeah. So if there are no questions, then I would like to propose a vote of thanks of the webinar. So Professor M. Suryakalavati Garu, Honorable Vice Chancellor of Yogamana University, Chief Guest of today's function, and the guest of honor, Professor D. Vijay Raghav Prasad Garu, Registrar of IVU, President of today's inaugural function, Professor K. Krishna Reddy Garu, Principal of IVU College, and the resource persons, Professor M. Hema Garu, Department of Virology, SV University Tirupati, and Dr. B. Pratap, Assistant Professor, RIMS, Kadapa, and then organizing team, the participants of the webinar, senior faculty, students, and research scholars of IVU, various academicians from all the corners of the India, and then ladies and gentlemen, print and electronic media, a very good afternoon to you all. I feel honored and privileged to get an opportunity to propose a vote of thanks on this webinar. I, on my own behalf, convey heartily thanks to today's chief guest, Professor M. Suryakalavati Garu, Honorable Vice Chancellor of Yogamana University, for her excellent message and benevolent support in permitting us to conduct this webinar. Thank you so much, Madam. And we are extremely grateful to our Dynamic Registrar of IVU, Professor Vijay Raghav Prasad Garu, for his remarks on this occasion and a beautiful explanation of the COVID therapies and vaccines and financial support for this program. Thank you so much, sir. And we are extremely grateful to the President of the program, Professor K. Krishna Reddy Garu, Principal of IVU College, for presiding the occasion and for the support and the encouragement and so on. Thank you so much, sir. And today we are very inspired to have eminent invited speakers like Professor M. Hema Garu, Department of Virology, SV University Tirupati, and Dr. B. Pratap Garu, Assistant Professor, RIMS Kadapa, for their inspiring lectures on the COVID-19 vaccines and therapeutics in this pandemic. Thank you so much, sir, and Madam, for being with us. And we are very grateful to the IT team who is doing technical services to IVU in providing the links and so on. So thank you, Professor Nazir sir and Anil for their support. And I convey thanks to all the other invitees, distinguished participants from Yogamana University and also SV University and Government Medical College, Kadapa, and then Fatima Medical College, Kadapa, and Jamia Hamdard University, Delhi for their support in participating in this webinar, and from various other degree colleges like from Kadapa, Anantapur and Tirupati and so on. Thank you so much for being with us and for participating in this webinar. And I thank all the faculty and then the research scholars, students, academicians, and non-teaching staff of the geology and genetics and genomics for their benevolent support for the active participation for this webinar. Not the least, very sincere thanks to the print and electronic media, PRO Dr. Subba Reddy Garu, Dr. Suresh and Bobilesu for nice coverage. And thank you one and all, and stay healthy and safe and have a great day. And before closing this webinar, I would like to request all the participants to fill the feedback form and also to fill the quiz also if they are interested. And I thank the student friends for active participation and learning during this pandemic. Thank you so much for all of you. Thanks once again, and thank you.